For my presentation, I did postmortem factors that influence meat tenderness. I've actually never taken a formal meat science course before, so a lot of the information we've learned so far in our course has been new to me. I really enjoy learning about the rigor mortis and some of the postmortem factors and how they affected the meat quality. So I decided I wanted to look more into the postmortem factors and how they influence tenderness, just as a more narrow category. So the factors that I chose was stress prior to slaughter, which that's like a pre-mortem type of factor, but it also has effects post-mortem. Electrical stimulation, aging, cathepsins, temperature, and pH. The first factor is stress. Stress prior to slaughter is thought or known to decrease meat tenderness. This is due to physical and chemical changes related to stress. They cause changes to color, texture, flavor, and tenderness of the meat. One of the main researchers in this area was Temple Grandin. She believed that animals fighting, electrical prodding, general excitement, high temperatures, and other stress prior to slaughter caused meat to be less tender. She'd spent a lot of her early career anyway, coming up with ways for gates and whatnot to be arranged in ways that were less stressful for animals prior to slaughter. An example that she had used in her paper was sheep that were injected with epinephrine 5 minutes prior to slaughter had carcasses that were tougher than sheep that had not been injected with epinephrine. Some contradicting ideas that I had found. There was a study done in 2014 that had some low stress situations and some high stress situations for some pigs. And it showed that low stress seemed to affect the carcass quality, but not necessarily the actual tenderness of the meat. So that was just an idea that contradicted what Temple Grandin had done, but her work was a lot earlier than what like this 2014 study was. The second factor that I chose to do was electrical stimulation. The use of electrical stimulation makes a carcass to be more tender. A carcass that is, these are some ideas that are already known, but a carcass that is already of high quality and tender will not benefit as much from electrical stimulation as a carcass that is not as tender. It does have advantages such as causing a carcass to be more tender, have better color, quality grade, and flavor. There was a study done that looked at high voltage electrical stimulation that had positive effects on tenderness, but in the study they believed, it was a 1992 study, they believed that other factors such as aging, calprotectin, temperature, and pH played a large role in electrical stimulation was not necessarily the main factor that caused the meat to be more tender, but they thought that the electrical stimulation played a role in the tenderness. Some contradictory studies that were done. There was a study done in 1978, which was prior to this study, but that said that animals with stress prior to slaughter, their carcasses would not benefit from electrical stimulation. In this study, it just showed that carcasses they didn't think it changed much when an animal had stress prior to slaughter. So they believed that if you had a low stress situation and then use electrical stimulation on this carcass after slaughter, that you would have a better quality, more tender carcass. My third factor was aging. This was actually the factor that I found to be the most interesting. It was generally thought that aging always increased tenderness. The factors like the species, the age of the animal, and the quality of the meat played a role as well. There was a study done in 2012 by King that showed that aging helps produce a more tender product, and had to do with it being a longer period of time. Some initial things and what we had talked about in class was that a lot of the aging was done for 10 days. In this study, they looked at doing 14 to 21 days instead of the 10. Some negative side effects they found was that the color change that occurred in some of the meats that had been aged longer was making the meats be less desirable. Some thoughts that they had from that study was that if they could find a way to pack the meats better, it may help the color remain the same while allowing the meat to age long enough to still be tender. Some contradicting ideas. There's a study done in 2016 that looked into ways of freezing prior to aging to see if that would help with things like color. But it ended up that study they believed it had to be like breed dependent. So the breeds of they'd used two different types of beef cattle. One was a Nellore, and the other one was an Angus cattle. And they found that freezing prior to aging helped with the tenderness, but that they found like more of the changes had to do with the breed of the cow, not as much necessarily the aging. And then there was also a study done around the same time that showed that breed differences affected tenderness in response to aging, but that there was other factors that played a role that included the gender. So heifers tended to be more tender than bulls. In this study, they used a couple more breeds. They used a Czech breed, Galloway, Simmental, Charolais, and some crossbreeds. So basically from that study, it suggested that aging as long as 6 weeks could be best for tenderness. But in this study, there wasn't anything mentioned in the way of color change. But I would imagine that if they were using 6 weeks, that there was probably some color change that would not be desirable. So in the study that had been done that looked at color change in the 14 to 21 days, these were the two charts, the graphs that I included in my paper that just showed the differences. And the days are up here at the top, and you can see the bars. The graph on the left showing color changes, and the graph on the right showing the overall tenderness. My next factor was cathepsins. In class we learned that cathepsins are membrane-bound proteases responsible for degrading proteins such as contractile proteins, myosin, and actin, and they work best in acidic conditions. There was a study done in 1988 that looked at cathepsins, and it looked especially at cathepsins B and H, and thought that they were responsible for tenderness, but they also believed it had to be related to aging. They believed that the cathepsins had a positive effect on tenderness. Some contradicting studies found that this was a study that was actually done in fish, not so much in like mammalian tissue, but just the idea that cathepsins might actually not make something more tender. This study was done, and it looked at fish, and it actually showed that the cathepsin activity in fish can actually cause the meat to be tender to the point it's unfavorable. And it had to depend on what cathepsins were being looked at, because the cathepsins that are active in fish are actually different from the cathepsins that we see in mammalian tissue. So I just thought that was an interesting difference. My next factor was temperature. Temperature plays a role in many different areas associated with meats and carcasses. So there can be the temperature like directly after the animal is slaughtered, and like effects we learned about like shortening and of the muscles related to the temperature afterward, and like when they're cooled down, and like hot shortening and cold shortening. So we learned about those things. Some of the studies I specifically looked at were more related to cooking and tenderness. So from the studies, it was found that cooking at high temperatures makes a meat more tender. Most of the studies all seem to agree with that. And the study in 2010 by Crawford showed that cooking at high temperatures makes the meat more tender. But during this study, they did some research looking at some different breeds of animal. They thought that had the bigger impact as opposed to the temperature. In both of these studies, they actually talked about temperature actually being, if it was too high, they actually used the example of 79 degrees Celsius. They preferred it to be 76 degrees Celsius for being tender, but if you cooked it too high, it would actually make the meat too tough, which most people already know from just general cooking. My final factor was pH. In 2014, there was a study that suggested that the high ultimate pH, which is the pH after 24 hours of slaughter, the study suggested that a high ultimate pH would lead to a more tender meat than that of a carcass that had a lower ultimate pH, so a lower pH after 24 hours. It also suggested that meats with low or intermediate pHs with aging would become more tender over time. So if they had a lower intermediate pH to begin with, if you gave them some time, they would become more tender. So generally, like in these studies, the carcasses were from around 7.5 when they started and would fall to be about 5. It was suggested that in all of these studies that temperature played a role on how the pH changed and what it did to the carcass. There's also a study that had already been done in 1996 that suggested that using aging based on pH would make a meat more tender. So in this study, they looked at different pHs, and then based off the pH, they were looking at the amount of time that they would allow the meat to age and what the effects would be. So in conclusion, after looking at all the different factors and the different studies that were done in each one, it was really interesting to see that how all the factors went together and how it made a difference in tenderness. And almost all the factors, if you were looking at one, all the other ones would come into play and would be important for each factor. Some of the factors I thought that would be the most interesting to study that maybe could make a big difference would be in aging. It seemed like aging played such a role on almost all the other factors, but then you'd have to make sure you kept things pretty much the same, like your stress, your pH, your temperature. But the only downside to the aging was that the color, after like so many days, you'd like lose your color that you would need. But maybe, like they said, with some different types of packing, they could make a difference by allowing the products to age longer. Though I'm sure there would be some downsides, such as the money of allowing those carcasses to age for so long. Some of the things that I was surprised about was that there was not a lot more research on cathepsins. For that one, there just didn't seem to be as much information as I was expecting. And the pH, like a lot of the ideas that I had gotten from the research, was that it was kind of a more generally like accepted type of idea that they already knew what was going on with each of them, as opposed to some of the other ones that seemed like there was more research done on them. And then these are all the studies that I used in my paper.